we get experimental science. We're curious, non-judgmental.
All right, y'all, we're going to have second guest on tonight. We are going to have Guppy on as well. We're going to be chatting a little bit more about that tonight, answer more questions. He's going to be talking to us a little bit more about the philanthropy side of Hutch as well, in addition to a broader series of questions about what kind of science is being done at the Hutch and answer these particular questions y'all may have. Guppy, welcome the heck on. Thank you so much for coming on with us and to collaborating with us and allowing us to join y'all on this fundraising mission.
No, Balint, thank you, and thank everybody in the chat for having us. Like, talking science is the funnest thing, so why not, right?
It's an absolute blast to be able to chat, and like there's so much variety as well in terms of like the research that we're able to chat about, and I'm really excited to have you here and chat with us, and it's been absolutely brilliant so far, so thank you again. Could you tell us just a little bit about yourself? Like, I'm super excited. Actually, our introduction has been I met you on Discord, but I'm like, I want to hear more about this guy. I want to hear about this, like, because you know so much about all these different topics, and I'm just like, I want to hear more. What does he do? Where is he from?
Yeah, so as some people have sleuthed out, my real name is Amitabha Gupta. My pronouns are he, him, his, but pretty much everybody knows me as Guppy, and it's not because of America that I acquired the name Guppy. I had the nickname Guppy back when I was in India as well. I happened to be in a class in 4th grade with 4 people with the last name Gupta, 2 people with the last name Amitabh, 2 people with the name Amit. So there was like really no room to move. And so I went with Guppy, and it just stuck. I manifested it to myself. And I came to the US. I was inspired to become a scientist when I read about the race to sequence the genome. And I said, that's what I want to do. I had one of my grand aunts had cancer, and I'm like, I want to sequence the genome, learn what it does for cancer. And that was what I wanted to do from when I was 14. And so I came to the US for undergrad. I did my study abroad at the NIH because, you know, I'm nerdy like that. And then I did my PhD in DNA damage and repair at Columbia, and I came to the Hutch to be a postdoctoral fellow, much like Pravrutha. But I realized that my interest was just like more. Like, science is about like those details and diving down. I loved the teaching. I loved the storytelling. I loved being able to see the big connections between lots of things. So I wanted to go on to become a teacher. And the Hutch said, instead of going to teach, why don't you stay here and teach about the science that we do at the center? I'm like, well, okay, that's a lot. But at the same time, it was so exciting because it's a lot, right? Like, there's so much to learn. And I started learning about what every single lab does. I started attending every talk, and you can start seeing how every piece connects to one another. And it is fascinating. It is being right at the edge of what people are discovering. And the other plus for my job, unlike Pravrutha, I have not had an experiment fail in 5 years.
Nice. That it's a humbling time when for 6 months nothing works. it's
6 months.
I don't know if you've had that.
You were luckier than I was.
we had one where we were looking at fruit fly ovaries and if they were popping or not, and there was construction above and below us, and none of the experiments all stopped working, and it turned out that it's because there was construction. The stress of the vibrations were causing their ovaries to pop, and that wasn't like what we were. So all of our experiments were off, and it took months and months to figure it out. It was like, oh, so
yeah. But there's also the times when a PCR, the thing that we're doing to amplify DNA, to make changes, to make mutations, like Pravrutha was talking about, it's been working for years and years, and suddenly it stops, and then you're a scientist, you change out one reagent, you change out the other, you change out another, and each time it still doesn't work, and then suddenly once again it starts working. You have no idea what was different from any of the last 20 times, but then you just take it and move on, and you say it was meant to be, right?
Yeah. And then do you go back and figure out what broke, or do you just like, well, you know what, the science god said it was fine, and we move on?
Yeah.
Yeah,
Don't mess with it. It's working.
exactly. I mean, and Smike's mentioned we had another incident too where the cleaning staff changed out the cleaning product they used, and apparently the flies
Hmm.
could smell it, and that was causing behavioral changes. And so we thought, someone had changed a perfume or a body wash, and we weren't using like the deodorant anymore to try to figure out what was changed. And it was like, oh yeah, the cleaning solution changed, and that's what they were detecting. I was like, no, why was that? So
Yeah.
yeah, it's just
Science is messy, right?
yeah. And Sparky Pugwash, thank you for the $10 donation. We are now up at 600. We're at $600, y'all.
Wow.
Blowing
I
past
mean,
that
thank
goal.
you, all of you. This is crazy.
it's awesome. The colony knows when they got to step up, and they always step up, and it's really amazing to be part of a community that values science so very much, and where we're chatting about science, donating towards science. Like, the support is really, really amazing from this community. Thank you all so very much. So Guppy, tell us
Honestly, yeah. Thank you.
a little bit about what you do on a day-to-day basis. So you get to interact with all the labs, but how does what is the goal of like the position? Is it science communication?
Yeah. This is an excellent question, right? Like, so I'm still defining the role because I'm changing it because it didn't exist, right? Like, so when I made the jump 5 years ago, ostensibly my role was to help scientists talk about their research to external audiences, right? That was it. But in my mind, a lot of times we were relying on stuff that had already come out and they'd lost the drive, and we were trying to like really micromanage what they say, and I wanted the authenticity that comes with the science. So I started attending talks, like every talk that I could find, to understand what the strengths of different people are, because different people have different ways that they tell stories and different things that excite them about their project. And so how can we rewrite an event or a conversation so that they're in their best space to talk about the science in the best possible way, right? Like, some people do great on stuff like this. Others, yeah, I'll take a quote from you and put it in a news article somewhere. And yet others, I will have you meet with a person one-on-one. And so as I did that, I started being able to help build better like cases for philanthropy of, why you should fund this because it affects this, this, and this. And here's how all the research ties together for the greater goal. And so I could elevate the science of basic researchers like Pravruta, because everyone knows the end goal is cancer. We can see what happens, but there's a connection all the way back. And how can we draw those lines and tell those stories and tell better stories? And so I help tell stories at the end of the day, stories about science for fundraising, for education, for events, and for things like this. So
Awesome. Are you up for telling us some stories tonight about maybe where
absolutely.
the research goes, and where I think the really amazing connection of working with y'all is that we kind of see where the donations are going, right? Like, what kind of research is it? And I love those statistics of like the $5 and the 100 test tubes. Like, that's an amount that's really not discussed, right, in a lot of these other charitable groups. And it's not that they don't do good work, it's just it goes to almost this nebulous pot, and you don't know where it ends up going. We also have a question about your t-shirt. Someone really likes your t-shirt, if you could.
Oh yeah. So this is from the human biology division at Fred Hutch. So one of the perks is I get to go to every division's retreat, and this is an animal cell, different than a plant cell because there's no cell wall, and there's mitochondria, so these little things, instead of chloroplasts.
It's a really cool shirt. I was like, oh, that's a really cool shirt. Nice.
So this is the nucleus. This is what Pravruta was talking about right before.
Nice.
So this is a nucleus of an animal cell.
And that's a wild type one because it's not fallen to the bottom, right?
Correct.
So you look at that, chat. We can look at Guppy's shirt and figure out the biology, if it's cancerous cell or non-cancerous.
Wait till I pull out my T-shirt that has a UMAP from large genome single cell sequencing.
No. Okay.
It's a joke UMAP that they made out of it, which is great, but I still got like free swag.
No, that's awesome.
Like science swag.
That's awesome. There's Yeah, are you up for telling us? Yeah, just like some of your favorite stories and research that has made
Yeah.
a difference. You feel?
Well, so I wanted to like start with like Pravurta's research, right? Like there's so much there, right? Like just that act of like those changes in histones causing the nuclei to fall in. And it speaks to me because I was in the lab next door to Harmeet, or like down the hall from Harmeet, studying cell division, right? And so that region of where the centromere binds, there's this massive structure that goes on top of it called the kinetochore. And the kinetochore is the piece that binds to the DNA and helps the DNA go one to mother, one to daughter every time the cells divide. And so as Proverta is talking about it, I'm like, wait, do the kinetochores load correctly? Is the fault that the cells can't divide? And in all cancer cells, without fail, you see errors in cell division because sometimes some will have way too many chromosomes, some will have way too few chromosomes, some will cut between other parts of the chromosome and mix and match, creating brand new like Frankenstein proteins that can then drive the cells to keep growing. So there's so many ways to go. Like if the kinetochores don't bind or the cell can't sense tension, then what will happen is the cells will start dividing without being properly lined up, and as it's pulling, it'll shatter the DNA, leading to that DNA damage that we see.
Oh
And
wow!
my PhD was in DNA damage, so it's like streams crossing. This is awesome.
That yeah, so it's all linked. It's that web that she was talking about, right? Like you
Right.
break one component of it, is that histone of how it's being plugged in. But like you were saying, if the binding's off, then that's gonna shear the DNA and break it further. And if the histones are misplaced throughout the chromosome, like she was saying, to its further possibility of that. It's really cool how it's all linked together.
Absolutely. And cell division is like crucial because cancer cells somehow divide faster than normal cells. And some of the breaks that are normally put in, some of the checks that are put in of like, is everything okay, is broken in cancer. And so, you know, everybody talks about, you know, the end goal is important, but for us basic scientists, it's like seeing how things normally work, poking it with a stick and saying, how does that break? And then tying it back to like the final end goal of like, oh, it broke this way in this cancer. So, yeah.
That yeah, it's like if you had a car, right, and let's like break a piece of
Yeah.
it and just see what exactly goes wrong. But here, I think one difficulty with cancer is that folks always, it's hard when you're studying how these end up working, right, is that it's not just a single cell population and everything is stable, but rather there's a tumor has a ton of different cell types, and there's also communication from wild type cells all within. And so
Yep.
how can that manifest, right, and how can you study it in a reasonable way?
Right. And, you think about it this way, right? Like cancer cells, regular cells are getting the same food, but somehow cancer cells are generating something else that's allowing them to divide. What are they doing differently? Are they getting different things out of the nutrients? What are they? And once you figure that out, you know that they've done a shortcut. They've made a change that has left them susceptible to that treatment. But how do we figure out what that change is? And that's what the basic sciences do, is you figure out how have you over leveraged yourself to gain that growth advantage. So now we can come in and target your weakness. Like, we have. I'll tell you one story. We have a researcher who studies metabolism, right? Like every cell in our body takes in glucose and goes through something that's awfully boring and complex called the Krebs cycle. Every one of us dogs, every biologist has learned the Krebs cycle, forgotten the Krebs cycle, relearned it. It's just way too many. It's how a cell takes in sugar and converts it into energy and other important pieces. And so it was thought that cancer cells eat more sugar because they need the energy to grow. But what one of our basic researchers did is he went and looked at which components are modified in certain cancers and figured out that the cancer was sort of short circuiting the Krebs cycle, not for energy, but for something else that would allow a different part of the cell metabolism to like make something different or grow differently. And so that exposed a weakness for that cell and how we can then target it.
So were they looking for that glucose change, or was that a serendipitous effect? Like, was it they were looking
No, no,
here
they were
and it ended up being
no, they were looking
elsewhere?
for that glucose change because so when I was in grad school there was, and I think it's still common now. There's something called the Warburg hypothesis,
Mm-hmm.
and it's that idea. So and this is what they do for like radiology to look for where cancer is. They put in radioactive glucose into your body, and you see the glucose clumping right around the cancer cells, and it's like because it's consuming all that sugar faster. And it's like, oh yeah, they're consuming sugar to get that energy to grow. But what he found was nobody had actually said, is it the sugar to the energy that is letting them grow? And he actually like went through and dissected the steps of what it was that it was doing.
So a question from Chad: is that why we're told to cut sugars from the diet as much as we could?
So those are two different things. So I want to so every single cell. So we are 3 trillion cells, maybe 30 trillion cells. Uh, every cell needs a certain amount of sugar. All our foods are broken down eventually to sugar in our cells. With regards to sugar in our diet, that's a different thing entirely. That's sort of the sugar we take in our system is broken down by our gut and our liver and by insulin, right? Like that, all of that. A lot of research is showing that like excess sugar in our body can lead to increased fat adiposity and sort of an increase in our immune system. So we get a hyperactive immune system. So we're in this state of inflammation. And inflammation, by and large, is one of the things that people can really easily tie to increased risk of cancer. So when your immune system is going wild and attacking a lot of other things, that's usually the cause. And so sugar is bad in that it leads to increased adiposity, increased body mass, and those things are the ones that are linked with increased risk for cancer.
So it'll be like the secondary effect of that, not necessarily the sugar, but everything that it ends up causing.
Correct.
Yeah.
Correct.
And let's see, another question. So we have a question from Coffee Rock. So as you were talking about the cell division, so unlike the rapid division and growth of, let's say, a newt's arm regenerating, the cancer has a rapid cell growth with altered DNA or excess.
Yes. So the cancer cells, something is off, and that's what the hard part is. A cell is such a finely tuned mechanism, and each part is responsible for regulating the growth. Why it grows faster in one thing versus another is entirely different. For some, they turn off the checkpoints for DNA damage, so the cell doesn't care whether the DNA is broken or not. Another one sort of lets it skip from one cell cycle to the other and just jump and uh, keep growing and overgrowing. So it is slightly different from cancer to cancer, which is why it's so hard, right? And it's there's so many different mechanisms that can lead to the cell overgrowth, if you will.
Yeah, so Coffee Rocks is just it's even further complicated because of the variety of cancers, and they're all acting
Right.
differently. So even making a generalization on that level that you think would be really really
Yeah.
broad and high up is not general enough.
Yeah. Exactly. Because think about there are some things that we often hear are associated with cancer. P50, P53, BRCA, those are DNA repair proteins. So those are the things that fix the DNA when it's broken. And those mutations basically, our DNA is constantly getting broken and it would go fix it. And if those are not working really well, then it doesn't fix the DNA correctly. And so it creates either hybrid proteins or turns something on, turns something off. And that cell then starts growing out of control. But also, likely, it has to suppress something for that overgrowth to happen and to bypass the whole DNA damage checkpoint entirely. So, and I think he asked earlier about variants of unknown significance as well. That's the other thing is that these the genes like P53 and BRCA are massive. The proteins that they make, they're massive, and so we don't know what every alteration does. Some some are linked with cancer, and some are linked with nothing. And so we have people that are also doing the research of what change could lead to cancer and which one can not as well.
Which again makes it complex because there's multiple, right? That if they're acting together which changes are the ones that are contributing, and as well as having like one versus another, and like how they're all interconnecting.
Yeah.
And it's a giant puzzle, and as Prabhu was saying, it's a web, right? Like she was saying, even though you're altering just one sequence, like it's going to have so many other downstream effects. And so with cancer, where you're altering large parts of the genome, and like you were saying with the kinetic cores and pulling apart the DNA, if you're damaging at random the regions of the genome, you're going to have even more issues that are going to take place. So it's really remarkable we're able to discover anything.
Yeah, and you add to that humans are messy. That it's not just like, we're one human being, but within our 30 trillion cells are also 30 trillion bacteria and viruses, which are our microbiome, that are living on us and in us, and our body has to find a way to live in harmony with those things. It is, we are messy.
Yeah, I mean, and that's internal microbiome, skin microbiome, like there's just everything, and there's just variable upon variable. We have a question from chat as well. It's like, even with the BRCA mutations that you pointed out earlier, wasn't it not about a couple of years ago that they found out that other mutations also were influence breast cancer, not just the BRCA that have been identified?
Exactly. And so, somebody was asking earlier about prostate cancer as well. So a lot of men don't think about BRCA, but BRCA actually also leads to prostate cancer. BRCA is also associated. So if you have family members who have had breast cancer and have had genetic testing, you should get yourself genetically tested to make sure you don't have BRCA as a man as well, because BRCA is also implicated in prostate cancer, right? But our researchers, so they're working with patients and patient samples and getting, they're doing some things called genome-wide association studies, where they're collecting genomes from people all around the world, and they're looking who ends up with prostate cancer, who doesn't, and then identifying what are the common elements. And like you were saying, Balint, it's no longer just one thing. Sometimes it's a signature. 6 to 8 different things have to be changed all at the same time for it to lead to prostate cancer. Or you have to have some from column A, some from column B. And so one of the things that our researchers discovered, that there's a certain group of mutations that can happen that can suddenly make your cell look like it has no BRCA, even though your BRCA is totally normal. Like the DNA repair functionality is gone, but your BRCA is normal, and it can be BRCA-like.
That's, which is kind of wild to think about, right? It's like that part is actually not the issue, but then it manifests as such, and well, then how do you address that question as well?
Yeah. And so Smikes is asking about, I think he means VUS determination.
Yeah, could you tell us what is VUS for other folks in the crowd?
Yeah. So VUS means variants of unknown significance, right? So we are talking about BRCA. Let's use that as an example because it's really easy to do. We look at BRCA in Balint and we look at BRCA in me, and you line up those two sequences. There will be differences between Balint's BRCA sequence and my BRCA sequence. And is one of ours more associated with cancer or not? We don't know. So there's currently one human genome, but we need to diversify and figure out what is the standard genetic sequence for people of different ethnicities and different origins. That's one big failing of our genomics work.
So just to pull us back, because I think it's kind of wild that of what you just said is the thing that we compare everyone's genome to is one person's genome. That presumably we are very.
I thought it was like 3 to 4, but yeah.
But
yeah, it is
like
all
right, like 8 billion people to essentially like 3 reference genomes. How would you ever know what is a meaningful difference? Like you said, if you sequence your gene and my gene, and we do against this one person, we're gonna be different from each other and that person. How do we know that that's a meaningful difference? Yeah.
Exactly. And so, what people start looking at are mutations that show up repeatedly in different people. And so one thing is you can do the long-term study where you say, we looked at all the mutations in BRCA and looked at the outcomes for all these people, and they didn't have breast cancer, but these mutations almost always ended up in cancer. So you can say, okay, maybe these are really strongly associated, but that doesn't mean the other ones are not associated with breast cancer. And so we have some researchers that are recreating those single nucleotide, and that's what the CRISPR technology has allowed, is to make those small changes for each VUS along the way, and figure out what happens and which ones lead to cancer. And so we can then go back and tell people, oh, this mutation, it was unknown, but we know now that it's less likely to lead to cancer. So it's
How
going
does
back and testing.
how do you know where to start with that? Because that's almost seems like a terrifying prospect, right? You have the whole gene, like
Yeah,
in this gene, and like you said, we've got a couple of mutation sites that could be of interest. But how do you pick where to? Because like we also said, a certain combination might be the thing that's important. So like, how does the
absolutely.
researcher know where to pick, or how many do they pick, and like where do you even begin with this?
So this is the beauty of what CRISPR has allowed us to do. So I'm going to talk about some work from Jay Shenduri at UW and Fred Hutch. So he's primarily UW, but he's also a Fred Hutch faculty member. He and a team from UW, in collaboration with others at Fred Hutch, were basically able to use CRISPR to introduce every single known mutation of BRCA into BRCA,
Oh wow! So like
and then
one,
study the outcome.
wow. So one
So
like
create like, go ahead, sorry.
no, just please. I'm getting excited. I'm getting crazy excited
Yeah,
about this.
yeah, exactly. So there's this technique now you can do with CRISPR called genomic tiling, where you can just go and like make a cut and then you put in a segment to replace it, and you can make these single changes all the way along BRCA, and then you pull them in, you put them in into BRCA, and you see which ones, and you put them into like quote unquote wild type cells, and then you basically monitor for which ones lead to overgrowth, and then you sequence and you can tie back, because it's barcoded, which tiling pieces led to that overgrowth. And in doing so, you start getting to the, okay, this one leads to this much overgrowth, and we can test a smaller subset, and you can go through and figure out like how much of a growth advantage it does. And believe it or not, Smikes, that's not a ton of grad students. That's like, genomic tiling has become so cheap and easy that people can uh, do it really easily.
So
Um,
is that something? Speaking of cheap and easy, is that something that the donations would also go to that we're raising funds today? Something like that kind of is something like that possible from a donation from a stream? Like how CRISPR, we've heard, it isn't necessarily easy, but like, is it so price restrictive, or will a donation make a difference in something like that?
Absolutely, right. Like, and I think what CRISPR allows, what the donations allow us to do, is we can sequence a whole patient sample. Like, it used to be back in the day that it would take years, and now we can do multiple patient samples overnight and do sequence either the whole genome or the regions that just make the proteins, and figure out what are the differences in there, and then back calculate from there and figure out what is the vulnerability of the cancer, to get us to that world of precision treatment. Because at the end of the day, we want people to get the treatment that's right for them, and not something that's just generic like most cancer treatments are, because then we can reduce the toxicity, we can reduce the side effects, and we can improve the quality of life for patients.
So that kind of ties into an earlier question we had from Coffee Rocks, which is asking, how does one get that BRCA testing? And presumably, like you were saying, if you were at risk or if you are diagnosed with cancer, you'd use that kind of testing to inform a very particular kind of treatment for a particular individual. Like, it's no longer the one size fits all kind of approach, but making sure you can help an individual.
Absolutely. And so we have genetic counselors at Fred Hutch as well, where where you can go and you can do your, if your family members have cancer or you have an increased risk of certain cancers, you go do the genetic testing. And it's a hard thing, because I was just having lunch with a friend today, and she was talking about, um, I don't know if I want to know, right? Like, would I want to know if I have this genetic mutation that could make me more likely to have Alzheimer's? And what I said to her, which is what I'll say to everybody here, is that genetics is one part of your outcome. It does not mean that you will definitely end up having cancer or Alzheimer's or whatever. We are a complex organism. It's a mixture of our genetics, our environment, and our lifestyle. So if you know that your genetics are slightly at higher risk, it's never you have a mutation, you will definitely get cancer. That there's no such thing as that. You have an increased risk of getting cancer. Well, you can do a couple of things to reduce that risk. You can change your diet a little bit, you can add a little bit more exercise, or you change other parts of your lifestyle, like you get screened more often. You go to the doctors to look for certain things. The doctor says, be on the lookout. If this happens, then come to us soon. So you can find ways to detect it earlier. Because at the end of the day, a cancer detected early is very treatable and has some of the best outcomes. And that's what we want.
And thank you, Dacum, for another $25 donation. Dacum added a note, because graduate students, easy, simpler, than with a grin. Thank you, Dacum, for that additional $25 donor. We're at $625, chat, crushing the goal. So, Guppy, with as you were saying, with like, we're wanting to have this specialized treatment for everyone, right? And as you were saying, there's ways to maybe reduce the likelihood of you getting cancer. But at the end of the day, I've heard the statement, and tell me if this is a correct statement that people have made, that eventually everyone's going to get cancer, because eventually there's going to be enough DNA damage in a cell. Like, if you live long enough, you're going to have that. So what kind of research is being done to, as you said, maybe help lower that risk, or even preventative screening at the Hutch that people might find really cool? And any one of these, I think we're gonna find cool, right? Because everyone's like super excited about all of it.
Oh, of course, right? And I'm learning about all of this in real time as well, right? Like, and that's the beauty of my job, is like, a dude with ADHD, a nerd with ADHD and photographic memory. This is like my dream. I am living my dream every day as I do this.
It sounds so freaking cool to be able to be on the front lines of the research, right? Ask the questions and be learning from the labs, and then disseminating that information to the world. Like, I'm jelly. That's really cool.
Well, but I mean, and I get to have conversations with people like you, which is awesome, and chatting with this whole group, which is awesome, right? Like, this is the fun part of my job. The other fun part of my job, I get to do so many fun bits. But the Hutch is doing a lot of work in a lot of these places, and you are right to an extent, right? At the end of the day, the mutations that will give rise to cancer or the overgrowth is a game of chance. If let's pretend you and I are going out, we're at the roulette wheel, right? And there's 20 numbers, and we'll pick one. We won't win that time. But if we play it 10,000 times, we will probably win at least a couple of times. And that's what it is you're just increase. The older you get, the more cell divisions your cells go through. Every time it divides, it's got a certain probability of something going wrong that will lead to cancer. But our human body is so well set up that we can also, our immune system is detecting abnormal cells and abnormal cancers and flushing them out, as well as because the cell is such a finely tuned mechanism that certain mutations are just terrible, and that cell will die. So not all terrible mutations will become cancerous. So there's lots of checks and balances that are put in place. So there's a few different ways that the Hutch is trying to mobilize this. So one is understanding sort of risk factors, finding out genetic elements that can be linked to certain cancers. And I'll talk about some work that we're doing for colorectal cancer, which is starting to show up in younger and younger populations, and is actually disproportionately affecting African Americans, American Indians, Alaska Natives. And so we are partnering with groups to start getting genomes and methylomes. So somebody asked a question about epigenetics. Methylomes from different groups to really understand the different changes that are linked with colorectal cancer. So the more we know about what can lead to colorectal cancer and develop those tests, we might even be able to go in with something as little as an aspirin and say this person has increased inflammation that leads to this, that leads to this. If they have an aspirin, they could reduce that inflammation that would lead to reduced risk of colorectal cancer, right? Like if you can find those really easily treatable ones, right? But beyond that, like, and I'm going to put a seed in this because we'll talk about this after this answer, is also discovering the microbes that are associated with colorectal cancer, because there's increasing evidence that at least 1 in 4, 1 in 5, 1 in 4, maybe even 1 in 3 solid tumors are associated with a bacterial virus. The question is, does the bacterial virus cause it? Does it propagate it? Does it shield it? What does it do?
Right.
But imagine a world where we can actually treat cancer with an antibiotic. Like that's
wild.
That
right?
is
like,
wild too,
yeah.
right? And so that's So one part is discovering the elements. The other is making better diagnostics. So I put in the chat an article for prostate cancer where one of our researchers is using machine learning and sort of computational models to detect prostate cancer and prostate cancer recurrence, or what the make and the model of that prostate cancer are from just a blood test. So we make the diagnostic easier, less intrusive. Nobody has to come in and get like a biopsy. We could do it in the comfort of our homes. Whether you live in like rural Oklahoma or in Seattle or in New York City should not affect whether you are able to get screened, right?
Because that makes a world of a
for.
difference if people even end up going in, right? If accessibility to
Right.
distance to the hospital, or heaven forbid payments and fees, like all those elements, right? And no one's
Right.
like, there's a lot of people that are left out from that.
Right. So that's the other element. And then finally, the treatments, or even trying to enhance the immune system to fight cancer. Because at the end of the day, your immune system is just going and picking out those micro cancers. Can we retrain it to take out the bigger cancers, the ones that it misses? And that, Fred Hutch, we talked about bone marrow transplant really early. So that was the ability to put somebody's bone marrow into somebody else that would cure the cancer. And that our researchers have discovered, a lot of it is driven by a certain type of cell called T cells. So could we reprogram the T cells to find and treat those cancers as well?
And one further complicating factor, right, is that if you have those T cells and the immune cells, there's that immune cell T cell exhaustion where they're overstimulated and they just turn off. And so it's not as simple, right, as just to like direct the immune system towards the cancer and everything's fixed. There's even more. And we just got another donation from Thug Shells. That is the freestyle rapper you heard at the beginning of the stream for our theme song, with another $25 donation. Miss Shells, thank you very much. I'm sporting her merch as well. I adore that woman beyond all reason. Thank you, Michelle, for those $25, madame. And her mom.
To what you're saying, right, Balint?
Her mom's last chemo
Sorry,
was
go
on
ahead.
Friday, so this is making a personal difference as well to a lot of our.
Raiders!
And we got a raid.
Thank you.
Raiders, welcome the heck in, everybody! Raiders!
Science stream, but we ain't bugging. We over here about to leave.
Hello, everybody.
What we got now? The tribes arrived. I promise that I have prescribed you vibes.
That is Queen Shell
But first, we
singing.
got our Giants. Shout out to the stream for the raid. That's Science.
Raiders, welcome the heck in for paleontologizing. We have the legendary paleontologist Danny Anduza coming on in here with the raid. Raiders, welcome the heck in. How was your stream? I want to know all the things. We got a special guest with us today. Dr. Guppy is here with us from the Fred Hutch Cancer Center. Actually, our second guest today. We had Dr. Pravrutha on earlier. Research scientist from the Fred Hutch Cancer Center, and we're raising funds today to help fight cancer.
Hello there.
Welcome the heck in, everyone. We've got Claire Burr coming in, the Lanina. Hello. Murph is coming in. Jody Fish. Catatoon, welcome the heck in. Great to have you here. Paleontologizing. Yes, we have our crossover as we're raising funds to fight cancer research. There is the link if anyone would like to join us for it. It is pinned to the top of the chat if you would like to join us. Catatoon, thank you for the follow. Welcome the heck in. If we hit certain sub goals, we will actually be raffling off tonight one of these glass beakers. Every single $25 donation gets you in the hopper to win one of these for a ticket. And if we hit $1,000, we will actually be doing another raffle for it, independent of it. Yes, no, we are fighting cancer, Cobra. We are fighting it, not supporting it. No. We're actually, what's really cool, Dr. Guppy was telling us where all the money is going to. So, for example, a $5 donation will actually go for 100 test tubes. A $250 total will actually go to a sequencing experiment to sequence a particular cancer cell line in a patient. And so you get to fight and learn how we're fighting every single element of the cancers via the donations that we're doing. And yes, we have Dr. PhD, I believe, right, Guppy?
Yeah, much to my Indian mother's disappointment, right?
So my Hungarian mother's too disappointed in me. We can be together in that front. And so Danny,
no,
go ahead.
Go ahead.
No, all you.
No, I was just gonna say, I often try trust me, I'm a doctor, but I will say that if something happens on an airplane, don't call me. I'm not really helpful.
And yeah, y'all. So we're just chatting about the science behind what is done at the Fred Hutch Cancer Center, how that translates to the research, the research to humans in particular to fight cancer, and where the donations are going to today. Danny, how was your stream, folks? If you don't know Paleontologizing, single greatest science communicator on Twitch, YouTube, and the internet. He's a paleontologist, does live field work, also does beautiful science content from his office, dinosaur Q&As. He moved to a brand new place. Danny, how's the new office? I hope it is amazing. And if there's anything you'd like to share, please let us know, my friend. Again, everyone, welcome the heck in. Please consider donating as well. See, I'm still waiting for a fellow plane passenger to shout, "Is anyone here a paleontologist?"
I mean, will that happen when your plane is being attacked by a pterodactyl or a pterodon, or what's the difference between the two? Is that the only time that we would call for a paleontologist on a flight? I'm not sure.
or all the time. All the time, you might. I mean,
or all the time, right?
Yeah. I mean, I know I'd love to call for a paleontologist at any moment on an airplane. I'd be like, what's going on? And for those coming on,
yeah, my wife is a geologist who started out because she was interested in paleontology, but then moved to just general rocks. So
what you're saying is that your wife rocks?
My wife, hey, yes, indeed.
Y'all, I am so sorry. That's. I mean, I'm gonna get the air horn for that. That's a pretty good one. Is anyone Genesis? Yes. Let me introduce myself to the Raiders as well. My name is Balint. I'm 1/3 of Science streams. The other 2/3 are my wife Lita and our daughter baby Alona. We're both research scientists. We are PhDs in molecular and systems biology. And we do interactive science streams here on Twitch, and we're proud to have partnered with Fred Hutch Cancer Center to do 2 charity streams in the month of December, and hopefully more as well afterwards. I'm chatting about cancer research, what that means to us as people, work and the research being done at Fred Hutch on everything from fruit flies to yeast to mice, and that information and how it's going to help us someday. So y'all, welcome the heck in. Please, if you have any questions, Dr. Guppy's answering them. I know we're backlogged on questions, but please ask. That's why we're here. And if you can and able to, please consider a donation, just like the very generous person who just threw in a donation. I believe that was Better Yet Shark. Better Yet Shark with a $50 donation. Message of, we're currently having to prep for a bone marrow transplant. Anything to help allow better treatments for cancer in the future. Shark, thank you very much for that $50 donation. We're at $700, y'all. Our goal was 1,000 across 2 streams. I don't want to get ahead of ourselves, but I think we might make it in 2 streams. So thank you all so very much for your generosity, and everyone, welcome on in.
And Better Yet Shark, really, really sorry to hear that, but I can tell you, I've met with and I speak with a lot of our doctors and researchers that are continually doing work to make bone marrow transplants even better and safer and more accessible for more people. It's come a long way, and I think it can get even better, but it is curative for many blood cancers and blood disorders. And actually, next week you'll be talking to somebody who's doing basic research that the outcomes of which leads to like myelodysplastic syndrome and some types of leukemia. Sorry, spoiler for next week, getting ahead of Elizabeth and her work that she's going to talk about. But yeah.
Yeah, and we have really cool samples to look at under the microscope for next week as well, y'all.
Yeah.
Jody Fish says, my boyfriend went through colorectal cancer at the turn of the decade. And we were actually talking about colorectal cancer right now, of like the potential preventative treatments. Guppy was giving us a little bit of a teaser about how the microbiomes, the bacteria, on our skin and around our cells, how that might influence colorectal cancer. And he was talking about the potential, and please expound on this, so one day maybe even taking an antibiotic to fight off cancer. And please tell us more about that if you can.
Totally. I'll tell you two different stories. So one is a diagnostic piece. So the microbiome, for all the new people, all the paleontologists here, you talk things big, we talk things small. The microbiome is the collection of bacteria and viruses that live on you and in you. And I was talking to a microbiome researcher about why the gut microbiome is so important versus how did the things get there versus the skin. And he told me something that just really blew my mind. He's like, well, actually, if you think about it, your esophagus and your gut is contiguous with your skin. It's all the same surface. That is your outermost membrane that is facing the world. So if you imagine, like, it is the part that is accessing most of the world, because you're eating, drinking, everything's going through it. And you have this collection of bacteria and viruses that live in your gut. And they live in harmony with your immune system, because your immune system has gotten used to them in some way, shape, or form. But there are certain bacteria and viruses that, through his research, has found are more associated with colorectal cancer. Like, people who have these 3 or 4 bacteria are more likely to have colorectal cancer. So he's developing a test to look for sequences of the bacteria in your stool rather than polyps or blood, to be predictive of whether your microbiome is off and is therefore likely to put you at increased risk for cancer.
And so that's a less invasive test, right? Like, you don't have to get a blood draw for that.
Yep.
You could just take it at home, and hopefully it'll make
You just poop in something and set it over.
That's awesome. And Legion 54, thank you for the $10 donation to Fred Hutch Cancer Research Center.
Yeah.
Thank you, Legion. Fully support smart people.
And I'll comment.
Okay.
I'm going to comment on what Sparky's saying. Yes, fiber. So we'll talk about fiber. I'll just say really quickly for fiber. Fiber is critical for your microbiome to eat. And so more fiber, better. For the average male, you should have 35 grams of fiber a day. For the average female, you should have 28 grams of fiber a day. It is really hard to get that much fiber in a day. And in an ideal world, you'd have like a diverse group of fibers, so not just the same thing over and over. So public service announcement, get 35 or 28 grams of fiber from a diverse set of sources: grains, fruits, more vegetables. Whenever possible, eat the rainbow. So as many different colored organic foods will give you the broadest spectrum of fiber. So, yes.
And we just got a $100 donation from Corn Relish.
Woo.
Corn Relish, thank you so much. We are at 810, y'all. We are at 810. Thank you so much, Corn Relish, for that donation. Oh my goodness, y'all. We are crushing it.
That's unbelievable.
We're at already 810.
That is unbelievable. So I want the other story that actually blows my mind even more with regards to like microbiome and colorectal cancer is the work that's being done by one of our researchers, Susan Bowman, and her husband, Chris Johnston. So she studies colorectal cancer. He studies head and neck and oral cancers. So there's this bacterium called Fusobacterium nucleatum, which is technically an oral bacteria. And it is associated with more head and neck and head and neck cancers. But with poor dental hygiene, it can go into your gut if it overgrows and it goes into your gut.
Whoa.
And when it's in your gut, it is associated with far more invasive and aggressive colorectal cancers, to the point where those cancers that are associated with this Fusobacterium nucleatum are more likely to metastasize and move to other parts of the body. So metastasis means when it moves from the primary site to other parts of your body. And so it's usually not the primary cancer that kills you, it's when it goes to the other parts of your body and takes over there. It's the metastases that mostly are the most dangerous things that to any person. So this bacteria is associated with an increased risk of the primary tumor metastasizing. And when it does, it also shuts down the immune system. And so the metastases almost take the bacteria with it when it travels. And so they're trying to figure out whether the bacteria is altering your normal cells to lead to it becoming more cancerous, whether it is shielding already cancerous cells from the immune system destroying it. So we were talking briefly about how our immune system is taking away all these microtumors in our body. Like it is basically providing something and shutting down the immune system for that, whatever microtumor which would normally get taken out to then grow out. Or is it changing something in the biology of the cells to move more freely or be undetectable? So they're doing some really cool work with that, and that is the next phase in our understanding of cancer.
I am blown away that there's a connection between oral hygiene and aggressive forms of cancer. Like, not in your mouth. Like, that is amazing, right? Like, can you pull back do you know the origin story of how that research started? Like, how does one get that kind of an idea? Because it's really important, right? You're telling us like the huge difference it's going to make, but that's such a cool initial idea.
Well, so I don't think it started out that way, right? Like it's it's just the idea of what are the microbes that are associated with different cancers. Let's just go find that. Because we've been sequencing people's cancer genomes for years and years and years. But a lot of times, you and I were talking very briefly about how there's a reference human genome sequence. So we sequence a person's tumor, we sort of line it up to the human genome. If it lines up, we're like, all right, it's here, it's here, it's here, it's this gene, it's this alteration. But when people are doing that research, if the sequence never lined up, it got thrown out. But what they can do is go look at that and be like, wait, does it line up with a bacterial genome somewhere or a viral genome? And then you find these links of like, oh, wait a second, the people with colorectal cancer have this sequence, which is from this bacteria. Or people with head and neck cancer have this, and you sort of, that's how you get to it.
So it's knowing what to pay attention to, right? Like, and not throwing out.
Right.
Yeah, which is... It's just such a cool story about that connection between those elements. I was kind of just shocked. I was stuck on that, and then how you were going the rest of it to like, this is just so freaking cool. We actually got another set of donations. We've got another $10 donation.
Wow.
From an anonymous one who says, in my world, cancer was present as long as I can think. I grew up with my family fighting cancer and my grandmother 3 times. Mother and aunt several times. Thank you for your fight trying to make a change for future generations. Corn Relish with the $100 and Legion with the 10 saying, I fully support smart people helping out the cause for everyone. Let's get it done. Rest in peace, Paul Allen, for such an outstanding person for his enormous support in this field. And Legion was saying that's an important philanthropist who helped with fighting cancer as well. So Legion, thank you as well for that $10 donation. So yeah, these I think one really important thing that you're telling us is also the preventative case of not necessarily just treating after the cancer and tumor have already formed and metastasized and left. And Coffee Rocks, thank you for another $25 donation.
Wow.
Thank you, Coffee Rocks. Y'all, we're 8:45 already. Coffee Rock saying, thank you, Dr. Guppy, for making this subject even more fascinating. We must win the war on cancer. Dr. Guppy, that you're crushing it, my friend. Thank you so much for coming on.
Well, I mean, I'll say, right? Like, the dream is that we shutter our doors or we go through a rebrand and no longer are the Fred Hutchinson Cancer Center. And someday we become the Fred Hutchinson Center for Infectious Disease Control, because viruses will live on long after we've taken out cancer.
Gotcha. That's a really, really good dream. An anonymous also with a $25 donation. Thank you, Anonymous. That's 870. 870 we're already at. Thank you, Anonymous. Literate Ian, thank you for the I did not know about this button here. Thank you, Literate Ian. Now we can see the live updates. I don't have to keep hitting refresh. So one thing that you've been mentioning that I think is really striking is that the war on cancer isn't necessarily to fight something that's already metastasized, but rather it's that preventative ability. And so could you, we started touching on it a little earlier on, but could we chat a little bit about the preventative cases that you can work on? So like the prevention that folks can do, and like the elements of that, and like what differences are being made on that front of like preventative care and testing ahead of time before, you know, not having to go to a blood test and all those other elements to it that we were talking about.
Yeah, absolutely. And you know, the person who was sharing that their family history, there's a long history of like they knew about cancer in their family for as long as they remember, right? Like for somebody like that, I would definitely say maybe go talk to your primary care, see if like genetic counseling is right for you, because if your family has had the same type of cancer, or figuring out what your underlying risk is with your genetics is really important, because that is definitely, I mean, it's not something you can do anything about yet. You know, the first gene therapy came online, was approved by the FDA last week. We can talk about that separately on another stream, because that's going to go down a whole different rabbit hole. But you know, what it's sort of knowing the cards you're dealt, and then trying to make adjustments to your lifestyle and your environment concomitant with it, and also knowing when to go get screened, right? Like first know your underlying genetic risk factors, know where that's at. The other thing that is lifestyle, right? Like, what do we eat? How much movement do we get? Like, there's enough data that doing a certain amount of movement every day is preventative for cancer. How much it is, those are hard things to study, because again, humans are messy. Some people have underlying genetic conditions, some people don't. Though we can't have someone exercise every day till they die, or till they get cancer, and then measure the amount of exercise. That's really hard to do. So those are really tricky studies, and similar with diet. And so that's why you'll always see in the papers like, oh, this thing causes cancer. No, wait, this doesn't. Oh, red meat does. There are certain things that we know for a fact, like increased consumption of processed food, red meat, increased body mass index. Those are associated with cancer, right? So those are things that you can try and control. And it's hard. It is hard, right? Like I often have to battle between, like, when I'm stressed, I just want that tub of ice cream, or that bag of Kansas City barbecue chips. Like I would eat bags and bags of that, right? But that's processed food. It's increasing my risk. I'm stressed, and it's okay. It's okay to do that, right? And it's about forgiveness and understanding where you can make the changes in your lifestyle.
So it's a balancing act then, right, on that front. It's not just
right.
None of the, you can't have anything. You have to be sad in life, but it's just knowing what risk factors you have and being able to take a reasonable amount of preventative pressures to lower that further, given what your personal risk factors are.
Exactly. And maybe you'll be lucky, like your genetic risk factors are like, yeah, you're all clean, just go on and live your life. I mean, there's probably something that we don't know about, but then sort of making sure that you're checking up on certain things. Like once you know your risk factors, you'll get certain tests more frequently, and making sure that when you hit a certain age, like I will in a few years, I will have to go get my colorectal cancer screening, being up to date on my screening, and making sure that those are being caught. Because like you said, if we catch a cancer early, the chances of curing it are really, 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 really high.
And that's where a lot of the current investment is happening now for that, right? To make sure that we can have better screening and preventative tools. Right.
And screening and preventative tools where we can screen and prevent for everybody everywhere, right? Like, just, you should not be able to live longer just because you live in a city with a cancer center, right? There are different cancers in different parts of the world. Sub-Saharan Africa has a very high cancer burden There's not that many cancer centers there. Their cancer types are different than the cancers we see. They have a lot more bacterial and viral associated cancers. Things like Epstein-Barr virus is a causative element for cancer in sub-Saharan Africa. Wow, holy smokes.
No, we
get
got another anonymous $100 donation. We are $30 away from our 2-week goal. Thank you all so very much for all the support.
This group is so amazing. Thank you, all of you. Like, this is just like blowing out the water of what we would really have expected, right?
I was like, I just saw it and popped. I'm like, whoa. We're gonna have to, as someone earlier said, we're gonna have to come up with some stretch goals. So we will absolutely come up with some stretch goals, y'all. We're absolutely gonna be doing that. Travel the World says my dad died at 58 of bowel cancer, so I go get a colonoscopy as directed by my doctor. Which I think Smikes mentioned earlier too. Is imagine that if you didn't have to get a colonoscopy, if there could be other tests that are less invasive that you don't have to,
right,
like be put to sleep for it, or like these, that at-home testing can make, I think, a world of a difference. And like you said too, Guppy, which I think was really important, and I feel in the field under talked about is accessibility to a cancer center or a hospital that has those kinds of tests, right? Like, it shouldn't just be for where you're geographically located if you're able to be protected, right? It should be everywhere. And so some of these tests are not geographically linked, right? Like, you could just send it to someone and they can take the test and they send it back and you'll know.
Well, and I'll give you a great example of some of the work that somebody like Jerry Radich at Fred Hutch has done on chronic myelogenous leukemia, CML. So CML is a really easy cancer to detect by a blood test PCR in the US. But it is very common in sub-Saharan Africa, but they don't have the way to do it. And so he, working with some other foundations, managed to devise a dried blood spot test so people can collect and send the dried blood to Fred Hutch and we will sequence it. And if we detect the cancer there, the foundation, along with the pharmaceutical company, will send that patient treatment for the rest of their life for that. It is very treatable if you can catch it.
That is incredible. And you know what else, Guppy? We hit $1,000.
Did we do it?
We did it!
Wow.
Dr. Cat Builds coming in with $30 to get us to $1,000. Now, chat, that means we have 3 things to raffle off. Now, 2 of those are going to be based on folks who've donated $25 or more. Counter Blood, what an appropriate name to come follow in. Thank you very much, Counter Blood. We are, Guppy, would you mind joining us for one of these raffles?
Let's do it.
So I know one of them was an instant raffle that we hit that was going to be one at 500, which our friend Kai Fish won, and one at 1,000, which we hit today as well. Oh my goodness. There was an additional 2 that we're going to raffle off, and that was for anyone who donated $25 or more. You'll be added to the hopper. We will do that at the end of stream tonight. So Dr. Guppy has a family as well, so he will not be with us until midnight. As I will be with y'all, don't worry. But we'll raffle those off at the end of stream after we have folks. If anyone wants to get in for a total of $25, you'll be entered in the hopper. We'll do 2 of those today. Dr. Guppy, what keyword would you like for people to use to get in on the raffle?
Ooh, this is a good question. I think, let me see, give me 1 second. I would, let's see, let me think I'm sorry. I will say, who was it that shared the story? I think we've been talking about colorectal cancer. We've been talking about getting tested. Let's do colonoscopy. So anyone who's 45 and older, get your colonoscopy. And the word for tonight is colonoscopy.
All right, colonoscopy, chat. Exclamation point, colonoscopy. Thank you, Smikes. Copy and paste Smikes' message.
Together, we can rule the galaxy.
And Peter, thank you for that resub from Karen Day is my name. Thank you, Peter, for thanking Karen.
Peter Steves just subscribed for 7 months. Thanks to @Karen Day's my name for my sub gift.
Thank you, Peter. Guys, exclamation point colonoscopy. That's a don't copy and paste it. Spikes wants the beaker. Don't worry, Smikes. We're gonna have at least 2 more tonight, and maybe we can ask the lovely folks at Fred Hutch if we continue raising funds next week as well. Maybe we can add in a couple of more beakers. Maybe. No promises, chat. We might add in some ancient amber for next week as well. So this Ubi is for anyone. This is for anybody. This is not linked to any amount of donation. This is just y'all can enter. We have 2 more that we're going to do at the end of the stream today. That is for if you've given $25 or more. I'll make a list at the end of the stream, add your name into it, and you will be also entered and will be pulled for the raffle at the end of stream for that, because I just want to make sure we give more time if anyone else wants to win one of those. So exclamation colonoscopy, will give y'all a couple more, maybe another minute to enter. I will need your mailing address if you win, just as everything else that we've been doing in the past in terms of raffles. Send me your mailing address via DM, and then we'll send it off to you. Pipettes, no, Smikes, no.
Pipettes.
That sir is.
Let me go chat with a few friends. I think some labs might just be replenishing their pipettes, but yeah.
Hey, look, that would be. I remember how much pipettes cost. So, but if they're getting rid of them,
right? Right.
If they were getting rid of them, then we'd absolutely take it. All right, last chance, y'all, to get in on that raffle. Last chance to get in on the raffle with the exclamation point colonoscopy as that keyword. I think Songbirder, thank you for that gift sub to Legion as well. Thank you, Songbirder, for that tier 1 gift sub to Legion. Legion, enjoy that ad-free viewing and the emotes made with love. Thank you, Songbirder. All right, y'all. Dr. Guppy, whenever you are ready, my friend, you tell me when to roll it.
All right. I think let's go for it.
All right. Here we go. Marianne. Congratulations, Marianne. Marianne, you have won the special beaker. Marianne, please DM me either on Discord or Twitch Whisper your mailing address, and we will get it shipped on out to you. Thank you, Marianne. And we got, remember, y'all, two more raffles tonight. That will be at the end of stream. That is for anyone who's donated $25 or more. If you donate, let's say $50, you'll get 2 tickets. You'll get 2 tickets into the entry. And if you donate $100, you'll get 4 tickets, and so on and so forth. So you can absolutely rig the system in the fight against cancer for the raffle for these really cool beakers. Thank you again, Dr. Guppy, for letting us derail you a smidge and just to raffle off that.
No, of course, right? No, this is. I'm so grateful to this community. You know, it can't be stated enough, right? Like, I think 95% of the donors to Fred Hutch donate $1,000 or less. It is a community, right? Like, fighting cancer takes all of us. It's not one. It's not. There's no magic bullet. It takes everybody, and we don't know which dollar for which experiment will find that critical thing for a treatment for a certain cancer or an early protection, right? We need all, we are grateful to all of it.
So maybe that's something that you can tell us about as well, is just how many active experiments do you think there are, like projects that are aimed at fighting toward this question, just at the Hutch, like just to give us an idea of how much research is done for, this kind of difference.
So what I will say now is that there's I would say conservatively about 300 odd researchers, PIs. So lab leads, there's about 300 of them at Fred Hutch, in addition to about another 350 to 400 clinicians who are seeing patients, working with patient samples, collaborating with our researchers. So I, honest to goodness, could not give you a number, right? Like, every lab has so many projects. Even if you start with 350 labs, many of those labs have at least 4 to 5 people who are each working on multiple projects. That number gets real big real fast. But what's the greatest thing about Fred Hutch that I've noticed is that people here love to collaborate with one another and work with each other, right? Like, it is they recognize that it is such a big problem. And more than any other place that I've seen for research, we have a very open door policy. Like, I could walk into any lab or any PI and just be like, hey, what's up? I had a question about this. And they'd happily chat with me, right? Like, and that's what you need. And actually, that's one of the benefits of the job that I've been able to do is I've actually managed to connect a couple of faculty that didn't know the other person was there or what they were doing, or that the projects had sort of, there was convergent evolution on two different projects, starting from two different places, right? And they could help each other. And it's amazing.
That's really,
Smike's
really
asked
cool.
about patents. We patent. We have a bunch of spin out companies as well, because companies are really a way to like get treatments out to patients faster. As a research center, we can't raise the funds quickly enough to get something out. So at some level, we do have to patent, license, and send stuff out.
Is there I want to also be respectful of your time, because like we have a family. I want to make sure we have Jimmy B. We got another raid in. Our lovely friend Jimmy B93. Hi, Jimmy.
Science stream, but we ain't
How are you doing, Jimmy?
Over here,
Music girl,
bubbling,
hello.
beauty, shagging what we got. Now the tribes arrived. I promise that I have prescribed you vibes. The vibes we got are giant. Shout out to the stream for the raid. That's science.
Raiders, welcome the heck in. Jimmy, how was your stream? Tell us all the details, folks. If you don't know our friend Jimmy B93, he's a besto friendo from the very original days of this stream. He's a gamer extraordinaire, a music teacher, along with his lovely significant other, Music Girl 95, who's here. Both of them music educators, making a difference in the world to all the kiddos. Super Mario 64 is complete. How was it, Jimmy? Was it a good journey? And folks, Jimmy is on the path to 1,000 followers here on Twitch. You get into 1,000, he's going to do a 36-hour stream, and if he has to take a nap on stream, he's going to do it on his cot. So guys, go drop him that follow. Jimmy, Raiders, welcome the heck in. My name is Balint. I'm 1/3 of Science streams. The other 2/3 are my wife Lita and our daughter baby Alona. We are research scientists, and we do interact with science streams here on Twitch most days of the week. And we've got a special guest today, Dr. Guppy, who is talking to us about all things cancer research at the Fred Hutch Cancer Center, and we're raising funds for cancer research. We hit our 2-week goal of $1,000 today for cancer research. Raiders, welcome the heck in. Jimmy, if there's anything you'd like to share with us, sir, you know the drill. Drop it in the chat. We will share the lovely Music Girl's throne list. And he went, Rah! There it is. Hit that throne list as well. But go buy Music Girl some stuff. She just got engaged, folks. She needs some items. Please support our friend Music Girl. Raiders, welcome the heck in. We were just doing a Q&A with Dr. Guppy. For example, Coffee Rocks asked if working with colorectal cancer, do you also work on pancreatic cancer? So I think maybe a broader question is, what are all the cancers that are researched at the Hutch?
So I actually cannot answer that, because if there's a disease area that gets a cancer, we do some amount of work on it. If nothing else, like the basic research that we're doing into the origins of cancer will affect so many other cancers, and they sort of cross tie in. But as Smikes is saying, pancreatic cancer is really one of the hardest ones. It's one of the cancers that has had the least improvement in 5-year life expectancy of any cancer over the last 20 years. And that's because there's a couple of different elements that make it so complex with pancreatic cancer. We have a team that is working on it, and we just recruited a new leader to sort of work more in the space of immunotherapy for pancreatic cancer. So with pancreatic cancer, what's really hard is that it isn't caught until really late, and so it's really hard to know the drivers of pancreatic cancer. And what we have one lab, the lab of Sita Kugel at Fred Hutch, who is diving into sort of recreating model organisms, model systems, to study the origins of many different subtypes. Because right now you get pancreatic cancer, you sort of get one type of treatment. It's thought there are 2 subtypes, but people don't know really, are there differences between subtypes, are there sub-subtypes? And so she's been able to create a whole different slew of models from patient samples to really understand what are the drivers for those. And to bring the story full circle, one of them is one of the pancreatic cancers is driven by this group of proteins called sirtuins, which are involved in histone modification and histone remodeling, which our stream started with Pravruta around. So histones are the proteins that DNA is wrapped around, and these sirtuins move them around to change the accessibility of the DNA to basically turn off and turn on some regions. And the lab of Sita Kugel has found that this class of genes is altered in a certain subtype of pancreatic cancer, making certain regions more accessible or turning on things that shouldn't be on to make them grow uncontrollably. So that does give us a direct thing of like, could we inhibit those? Could we target that class, that gene? What is turned on? Can we inhibit that to prevent that pancreatic cancer from growing? So, yeah.
Do you know by chance if these model organisms that are being studied, are those mammalian, or are there a bevy of other ones that could be?
No,
used?
so this is these are they're called patient-derived xenografts. So we're taking patient samples and recreating a 3D model system, because traditional cancer research has been done with like, you take a cancer cell, you disaggregate it, you put it in a plate, but that's not how it grows in our body. And so they're doing some work. They've managed to recreate it in like 3D space to really understand the geometry and the sort of structure of the tumor itself. So it's patient derived. It's recreating it in a dish that is 3-dimensional, and working with other researchers to really study what is the immune makeup. Are there certain immune cells that are in that cancer versus not? Because Balint, you mentioned like with immunotherapy, you want to know whether it's like getting exhausted or not. Here it's like, are there any immune cells at all? Are they exhausted? Are the wrong types of immune cells preventing the good type of immune cells from coming in, and starting to get at those questions? So
you get to get the whole image of the, like, it's not just, again, one cell type that you have in a dish, which is how we study some cancers, right? Because you have to start somewhere.
Correct.
But this is the entire microenvironment. Non-cancerous cell types, cancerous cell types, you get pretty much everything into this system. And then you can hopefully figure out a lot of the changes that occur, and maybe even try to test and see what stage it's at, right? Because that's going to be also really difficult to do.
Absolutely. And so figuring out, like, are there identifiable genetic drivers that we can then work on for the different cancers?
And presumably those will be individualistic as well for each patient, like you were saying, like how to best
at the end of it. Yeah. Because I think who was it who said it? Sorry, go ahead.
No, just I want to make sure that we're respectful of your time. So if you have a hard out at any point, please let me know. I'm gonna get carried away if I don't know what time. And I know Dr. Guppy's got a family and dinner and like all these things. So I want to make sure we're respectful of his time. So whenever you need to go, or just let me know so we can make sure that we don't keep you far over there.
Yeah. I think I'll give you like a 15-minute. So this can be my 15 minutes heads up, because I still have to go cook dinner as well, because my wife's gone to pick up my daughter from kindergarten. It's hard to believe. So
congratulations.
I made it through preschool, right? It is hard, but it is great. Raising a kid who I can now also lecture incessantly about different things.
Mine still just sits and listens. She just hit 10 months not too long ago, so we just she's in my lap. And
oh man, 10 months is awesome. I remember, so between like it's the first 3 months they're just like formless blobs, and then between like at 4 months they start having faces and they start reacting, and then at that 6 to 10 month when they're the potted plant where you can just like plonk them down and they're watching and happy, but they can't move and cause damage. Best time ever. But once they're moving, you will find out how not baby proofed your house is.
Yeah. So finding out that if you vacuum every other day, it's still not enough vacuuming because she's zipping around everywhere, and it's like, no, little one, where are we going? We go fossil hunting sometimes live on the stream, like we do IRL stuff. And she had this little cart that we were pushing her in, and she was just sitting there. And one of our second stream doing that, she actually stood up in the cart, which was, we caught the first time she ever stood up. We caught live on stream and got to share with the community, which is amazing. But the most recent time we went on Saturday, she would not try to stop trying to stand up and trying to get out of the cart and like jump in the river.
Mm-hmm.
It's like, oh my god, we're moving past that potted plant stage.
Yeah, and that then it gets real interesting at that point.
Yeah. But so with the 15 minutes, where would you like to take us? What would you like to do with another story, perhaps, of the research, and then maybe rounding out with the big difference that Fresh Hutch makes? Or we also have had some requests for you to come back. I don't know if you'd be willing to come back next Monday as well. No pressure.
Let me check my schedule. I think I can probably figure it out. Like I'll talk to Elizabeth and see how long she's on, and I can be sort of the chaser to the shot that is Elizabeth, because she's going to talk about something really cool, and that's I cannot wait for her to be on. But yeah, no, I'd absolutely, and again, like, we should keep talking. It's a pleasure to talk to this community that is so engaged and has so many questions about science that, it is invigorating for me.
Yeah, I can't tell you how proud I am of this community, that what we've built, and folks are just genuinely excited about science and learning, and it is a safe place to learn. You can come be a nerd, and it's good to learn. It's just, I'm really proud of it. It's not, you don't see it often, but I just can't tell you how proud I am of every one of our colony members here.
So let me see, where do I finish out? We've done a lot of talking about colorectal cancer and so sort of microbiome. So I'm going to finish with like a story about a new faculty member that we have that's doing some basic research, which is around this, that blows my mind even more. So like, as much as we're talking about like microbiome and colorectal cancer and that blowing your mind. So think about it this way, right? We were talking about how your gut is basically contiguous with your skin, right? And so it has to be a barrier most of the time, because you don't want random bacteria getting in under your skin into your gut. But at the same time, when you eat, it has to be able to absorb nutrients. So what is that switch from when your gut goes from becoming barrier to open door? What happens at that moment in time? And so we have a researcher named Jimmy Talbot, and was doing some just basic work trying to figure out, like, does it matter what he was using a mouse model system. He's like, does it matter what it eats? Like, is it the density of the food? What is it? And then he did a test, a control with like glass beads that can't be digested. And what he found is that it's not a food or a nutrient or anything that triggers it. It's just that mechanic. So the glass beads, if it provides enough mechanical tension, there are enough neurons in your gut that sense it. So the first brain that ever evolved was around a gut. This is technically our second brain. We have almost as many neurons in our gut as we do in our brain. And so they sense that tension. And at that moment in time, that tension tells the immune system to stand down. Those neurons are connected to certain immune cells that then secrete this protein into the environment that makes all the other immune cells stand down. So you're no longer at a barrier, and it changes the absorbency of your gut. And they went and looked at sort of not cryo-electron microscopy, but like I think scanning electron microscopy, like really fine microscopy of the gut. And you can see when the gut is empty, you see the microbes, which normally grow as like strings, these long filaments, shaved down to nothingness. And then when the gut is full, you see strings and strings and strings of microbes. So the takeaway I took from that is, when I'm drinking my kombucha and I want those microbes in my gut, I am doing it on a full stomach and not an empty stomach. Because if I'm doing it on an empty stomach, my immune cells are eating up those bacteria as fast as they're coming in. But if I have a full stomach, it's ready to absorb, and those bacteria can latch and then grow on out.
Wow. We actually, so it's really cool that kind of research is being done with like the proto brain, because we actually, so we do Science News of the Week here, and there had been a study a couple of months ago about that in a sponge, like being that proto brain is like the filter feeding, right, and like the neurons that are connected, and it's really cool. Like, we always hear about the gut microbiome interaction with the brain, but this is like next level stuff that you can see on an image as well. That's remarkable.
Yeah.
And it's the
It is
glass bead
crazy.
experiment is super elegant to be able to do it like that.
No, but it was the control. He didn't think he would get anything from it, and just the fact that the control was sufficient, because I think he was testing like different foods. What, like, do you need sugar? Do you need like protein? What is it that triggers it? And it was none of those. It was just like tension. So if you just like push it, yeah.
So what's the naively, what's the actual control for that experiment?
So the control was, so basically he was first putting in like just different food groups, and then he put in like glass beads, and as soon as the stomach was full, that is when he saw like the absorbency go.
Yeah, so like now would he, because presumably he's following up on it, right? Like what is the next,
Correct.
like the current control for that? Is that just like no tension, so just an empty stomach, I guess?
So there's no tension, so that's the negative control, and then the positive control is the beads. But then he has like variations in like sort of thickness, density of different liquids, and sort of using that to fill a stomach. Like,
so there's different amounts of tension that you can create.
Correct.
With that's
Correct.
really cool. That's,
and then
wow.
you start teasing out like which immune cells do I take away, which neuronal signaling markers do I take away, right? That you can start, so then you can start really disentangling like which neurons are actually sensing it, and what are they talking to then shut them down, and then which cytokines or like immune stimulating or immune suppressing molecules are present that we can then do something about.
Is it done on a dish? Like how like, can you just give us the scenario of how you're able to test this? It's not in someone's stomach, right? So like what do you do?
It has to be done in a living organism, so it's in mice technically.
Gotcha. So is it, you have to feed the mice the glass beads and the different amount of viscosity in the full stomach and the empty stomach, and then presumably take the cells and look at and do sequencing or look at them under the scope. Gotcha. That's
Correct.
also quite interesting that we can't yet model it in a dish.
Yeah, I mean, because the hard part is, right, like there's so many different elements to your gut, right? You can't rebuild a whole GI tract and the neurons. Like usually you don't get the neurons when you sort of do biopsies. I don't think, like, but maybe, I'm not sure. Like whether you could do it.
The similar issue, like where you have a bunch of different cell types in the tumor, and like having just a singular cell type isn't going to give you the answer. So presumably, even if you did have gut cells and you could put tension on them, all the neurons and how they connect, and the other cell types talking, and you said like the immune cells coming in, it's just too much, and you have to be doing it in a living organism. That is a remarkable, cool accidental study, right? Of like, this is our control, and it's oh, wait a minute, it's still doing its thing. Like, what exactly is the signal? That is amazing.
And then just think about that with regards to, we're talking about how your microbiome is associated with disease or not. The next phase is you alter the microbiome. So this will hopefully inform when and how you eat to then be able to alter that microbiome, right? That you can't just like throw something at it.
And from Chad, is there a connection between that and why you should maybe take certain medications with food, presumably because maybe you have to stimulate the gut?
So I think sort of, I don't know whether I can make a blanket statement on that one, but I think with food, like I think it's possible that like the you sort of need a full stomach because the drug, whatever it is, has really harsh effects, and you sort of need something to absorb, or absorption is better of the drug with a full stomach versus an empty stomach. I'm not I have not read up enough about that of
Gotcha.
why some things are better with a full stomach versus
Gotcha.
empty
It would be
stomach.
interesting. Maybe it isn't known yet, right, about those tensile forces, and maybe
Absolutely.
that is contributing, but we just don't know yet.
Absolutely.
I'm
And I think, yeah, we getting better about like how we link our microbiome and how we eat and when we eat.
which
with
there's an
what we
absurd
eat.
amount of links to it, right? There seems like every other day there's like a
Yeah.
new study on it that we didn't account for in the past. Same with like organism research organisms, like how diet's affecting behavior, neuroscience, the transcriptome, and then you see
Totally.
right papers of like, I know in flies it's say flies were fed standard fly media. It's like, what is your recipe different? Is that going to affect your results? And it's,
Oh yeah.
it's like Mike says, the more you learn, the more questions you're going to have, and yeah, it's the beauty of science.
Totally.
Right?
And so actually, you touched on something which is like, there was a new paper from a fly scientist at the Hutch, Akila Rajan, who basically found like that flies that eat more fatty foods, there is a connection between that and the brain, and it sort of leads to sort of decay of certain neurons and glia in the brain.
Huh. So the more fatty.
Maybe
it's
for the
like,
next time's chat.
Okay, there we go. Y'all, teaser.
Actually, you know what? Maybe I'll put that paper in the chat. Hold on, give me 1 second. But I mean, if this is interesting, I can absolutely find the person who did the research. She's pretty cool, and see if she would be willing to come on and chat.
Yeah, absolutely. I think folks have been really enjoying talking about the front lines of science and all that's being done at the Hutch. Is there any? I know you got to skedaddle, my friend, but is there any closing remark that you'd like to make before signing off for today?
The only closing remark is an emotion, and it's gratitude. And I'm absolutely grateful to you, Balint, and to Science for hosting us. And I'm absolutely so grateful for this community for welcoming us all today and being so willing to engage and have this conversation, and being so generous with their donations and helping Fred Hutch. We are in this together. So thank you.
Thank you very much for your time, for chatting with us, for taking a chance on us, meeting at TwitchCon, and we're like, hey, let's make this happen, and we sure enough did. Y'all, we have we're going to continue streaming tonight. We're going to continue doing some microscopy, raising more money tonight, of course. Next Monday, we're going to have a second guest, and Dr. Guppy's going to come back again, possibly on next Monday as well.
I'll work on my background next time. I should have planned that a little bit better.
It's weird that it doesn't blur, right? It's like every other service blurs your background, but this is the one that doesn't.
I know.
But yeah.
Well, I'll figure it out.
And if y'all are into it, we'll do additional crossovers as well. I'd like to do, a mission of our channel is at least once a quarter to do a charity stream. I think our value systems align so well that this is, I think, the perfect partnership to have. So thank you so very much again for your time, and we're looking forward to next week.
Sure. See you guys next week. Thank you so much again.
Appreciate you. Thank you, Dr.